collaboration since. Without further ado, this is going to be kind of in two steps. First, we're going to talk a little bit about stroke prevention and AFib, and I promise I'll not make it too into the weeds clinically, and talk about how I think we can leverage existing data to improve how we treat patients, and some hopefully very exciting work we're going to start. And then we'll talk a little bit about sudden cardiac death, and where I think we also can do better with what we already have. And so it's really going to be a tale of two cities here. I'll give a fair amount of background. I know everyone here is involved in cardiovascular research, but you may not spend all your time thinking about some of these topics clinically, and so I'll try to give you some background. So the first topic I want to spend time chatting about is how do we better risk stratify patients with atrial fibrillation in terms of stroke risk. And this has been a long-standing interest of mine. So atrial fibrillation, many of you may know, but the scope of the problem is the most common adult arrhythmia we deal with. Many of you study it on the basic translational side. This is one slide demonstrating the prevalence or the expected rates of AFib by the year 2050. And then another very similar type of study just by age group. As we all get older, our risk of AFib goes up. And when I see patients that have any cardiac disease over the age of 70 or 80, if they don't have AFib, I tell them they've won. So I had a grandfather at 93 that had heart failure and a pacemaker and never really had much AFib. He won the game as far as AFib is concerned, because it's just, it's not an inevitability, but close for many patients. It was recognized very early as part of the Framingham study that AFib increased the risk of stroke by a significant margin. Those patients were relatively unique compared to what we see today, but nevertheless, this was the seminal observation. And strokes related to AFib are particularly nasty. Stroke is the leading cause of death and disability, and strokes related to atrial fibrillation, the pathophysiology of which I won't necessarily get into today, but these specific strokes are particularly debilitating, in part because of their pathophysiology. So this led to the use of oral anticoagulation to reduce the risk of stroke. And this is a Danish nationwide study of events, basically looking at the reduction in stroke with the use of oral anticoagulation, OAC, and the concordant small but increased risk of intracranial hemorrhage, which is the most feared bleeding event or side effect or adverse event of blood thinners, of anticoagulation among those treated. Point of this slide is to highlight that we use these drugs, but it's under the in the effort of balancing risk and benefit. So for our relatively lower risk patients who have AFib, but not a lot of other risk factors for stroke, we don't necessarily recommend blood thinners. And as your risk of stroke goes up, we get more aggressive with them. But we're not particularly precise at this. So this is the seminal kind of data on risk stratification and what we use currently as the standard of care. Two scores, the CHADS-2 and the CHADS-VASC, again, may be very familiar to many of you. What I want to call your attention to are two things. One is the relatively small numbers by cardiovascular research standards. In other words, if you're looking to risk stratify and understand risk in a population, and you're going to make a guideline and just make treatment decisions based on it, these are relatively small single studies. What I also want to call your attention to is kind of the increasing stroke rate as the scores go up, and the rates kind of in the 1 to 2 to 3 range, which is really where we start making decisions. So as your score goes from 0 to 6 or more, with each of these two scores, the risk of stroke goes up, and our aggressiveness with oral anticoagulation goes up. So this has led to the current guidelines, where if you have a 2 or more in men or a 3 or more in women, because we no longer really recognize female sex as a risk factor, but this is how they decided to write the guidelines, you have an indication for a blood thinner. So that's kind of the background. And it's led to this kind of current state of the science. And science is a very generous term for some of us. But essentially, if you have a high score, which is a lot of patients, you're well represented in a lot of our trials, and you derive benefit from anticoagulation, and the benefit usually outweighs the risk. If you have a rather low score, it's unlikely you're going to benefit. But some of these patients still have stroke, rarely. And we are going to have newer and newer anticoagulants that probably have a lower risk of bleeding. And so the risk benefit profile may shift lower and lower. And then we have this group in the middle where we really don't know what to do. And they're not very well represented. There's unclear benefit. And so there's a gap here. But overall, our prediction is not great who to treat. And so there are several unmet needs. Number one is risk stratification is imprecise. My patients at high risk may never have a stroke, and I may categorize somebody as intermediate low risk, and they may have a stroke. The diversity of populations that are studied in this realm, especially upon which those guidelines are based in that risk score, is not diverse. There are always new and emerging risk factors. You may have heard of left atrial appendage shape, fibrosis on MRI, biomarkers in the blood, EKG characteristics that portend a higher risk. And getting from an associated risk to we should include that in our treatment pathway is a really long trip. And so we haven't gotten there yet from any of these. We don't currently acknowledge in any way, shape, or form any sort of synergistic or dynamic interaction of risk factors. So if you have high blood pressure and heart failure, you get a score of 2. If you're female and have high blood pressure, you get a score of 2. If you have diabetes and high blood pressure, you get a score of 2. We treat them all the same. And that is in part because historically, in order to do this in the clinic, you had to keep it simple, or else, in the midst of 40 patients in clinic, I don't have time to spend 20 minutes creating some risk calculation historically, especially back when we had paper charts. But nevertheless, there's not much synergism. And there are well-described disparities in the diagnosis, the management, and the outcomes of patients with atrial fibrillation, which the risk scores don't do anything for. And so these are the unmet needs that led to some interest on this campus of exploring this. And these are kind of the aims we are intending to explore with the methods I'll talk about and hopefully discuss with you all. So primarily, we're interested in developing a portable, so from healthcare system to healthcare system, more personalized risk stratification tool to improve stroke prevention or stroke risk stratification among patients with AFib. We think we can discover new risk factors that drive stroke risk. We want to develop a electronic decision support tool within an EHR that's more informative and dynamic. And plan to compare that to the current standard, and in part to improve upon it and to also address this and quantify some of the disparities and hopefully improve on them. So where did this come from? So this, in a large part, came from work done by Mark Yandel's group, who many of you probably know, over in bioinformatics and the Center for Genomic Medicine, and a great paper they published last year looking at what they described as Poisson binomial-based comorbidity discovery. It essentially is a way of taking all of the healthcare data in our health system and using it to understand risk and what drives risk at any certain patient population. And I'll describe the method a little more in a second. But it's essentially a way of leveraging all of the healthcare data we have that we collect clinically to understand disease risk and outcomes. It allows for automatically controlling for confounding in a kind of more sophisticated way, and a more powerful way of discovering what covariates influence disease. And so this is one kind of comorbidity map that was generated as part of these analysis. This happens to be for heart transplant in the middle, and what other disease states or medicines are closely linked with that outcome. So it's a little difficult to read here. I apologize. Cardiomyopathy, for example, congestive heart failure. These are all terms in the health system record that are obviously linked to heart transplant, some more clinically useful from a risk stratification standpoint than others, but was used to highlight the potential in the methodology. And this is one way of understanding perhaps novel risk factors, but what we really also want to do is understand, quantify risk, if you will, based on either new factors or factors that we're aware of. And so they can take those data and select their candidate variables and create graphical models and calculations for absolute risk and relative risk with or without certain, I'll use comorbidity very broadly here. So comorbidity being really any cofactor. So this leads to what they and I would describe as an explainable AI approach. And again, I'll get to this, a little bit of the math in a second. And can kind of capture synergistic interactions, which I think is key. So, so these are univariate. So milrinone, and many of you may know, is a medicine used to treat heart failure. Cardiomyopathy, obviously, would be a broad term. Again, this is for demonstration purposes only, may or may not be clinically helpful for this example. But what's important is you see these independent risk factors, and then when you combine them, the sum is more than the individual sum of the parts. And this at least demonstrates kind of the promise and the idea of getting at synergistic risk factors, which we don't typically do clinically. And then as part of the health record, and we'll get into some other opportunities for looking at this, we obviously have a number of covariates that get at disparities, whether it's race, ethnicity, religion, marital status, income, zip code, occupation, et cetera, which aren't necessarily classically included in risk stratification. Okay, so this is the relatively simplistic understanding of the binomial, the Poisson binomial approach. This is kind of how Dr. Yandel helps people like me understand it, frankly. But it's the idea that in a regular binomial test, if you flip a coin, each coin is treated as having the same risk of heads or tails. The translation is every patient, you treat as having a similar risk for one thing or another. And what the Poisson binomial approach allows for is every individual patient to have their own risk based on the cumulative data. And so you can vary the individual risk approaches, and it works a little better for patients than coins, but I think at least highlights some of the idea and the benefit of the Poisson approach. So we have all this data, we have this approach. How do we translate it to stroke prevention in atrial fibrillation? Well, the first thing they did as part of a subsequent paper is just compare significance levels. So these are p-values for different risk factors using classic binomial statistics and the Poisson binomial approach. And the red cells are reach statistical significance based on the Bonferroni adjustment. So you'll notice they're all under 0.05, which is the non-Bonferroni threshold. But essentially, when you look at different risk factors, whether it's age, sex, ancestry, ethnicity, insurance, span is how long they're in our health system. You are able to identify, even with smaller total patient samples, much more significant relationships using the PBC approach compared to a classic chi-square statistical approach. And so I think highlights, again, the power of this approach. So we endeavor to take it a step further and have submitted a grant that hopefully we'll get to pursue here. And looking at, okay, so we think this can do better. How are we going to kind of demonstrate preliminarily that this is promising and we should pursue it? So we came up with relative risks in different settings. And these, again, are preliminary data where we took that score, that CHADS-VASc score, which includes things like diabetes and high blood pressure and heart failure and sex and vascular disease, and tried to dissect it in terms of the PBC approach in our health system. So on the left are kind of the classic some of the classic CHADS-VASc risk factors and the relative risk of stroke with or without them. So diabetes, not surprisingly. Fortunately, our data are consistent with many international cohorts that have found that female sex is not particularly on its own a risk factor. But you'll notice relatively different relative risks, different relative risks among these different risk factors that each generate 1 point risk in the score. So in the score, they give you similar incremental risk, but in actuality, they don't convey the same risk, and probably don't convey the same risk for each individual patient. One person's hypertension probably isn't the same as somebody else's. Perhaps even more important is when you combine them, the example I highlighted earlier for a random score of 2, for example. So if you're female and diabetic, female and high blood pressure, heart failure and diabetes, heart failure and vascular disease, these all sum to a CHADS-VASc score of 2. As I hope you'll appreciate, the relative risks are quite different. And you would rather at least from a risk standpoint, almost have heart failure and vascular disease and high blood pressure and diabetes, at least statistically speaking. So we think this nicely highlights the limitations of the CHA2DS2-VASc score and the promise of alternative methods based on our data. And there's a ways to go here. On the right side of the panel is integration of some of the social determinants of health, which classically are not included, at least clinically, in our risk stratification of patients for stroke and AFib. And so it includes things like African American race, age over 65 is a CHA2DS2-VASc score, American Indian, I think, is AI, Asian, Caucasian, Hispanic, et cetera, et cetera. And as you'll notice, again, relatively different risks for stroke based on these. And then we can look at different distinctions. So patients that are both Hispanic and have diabetes versus those that are Hispanic without diabetes, isolating perhaps the effect of ethnicity. Similarly, we can look at payer or another a marker of socioeconomic status or social determinant of health in terms of a patient that has Medicaid versus not. Pacific Islander versus no with or without Medicaid. And then, what's intriguing here is age over 75. Most of those patients have Medicare, and if you don't have Medicare, it appears that your risk, at least in our data, is higher. These data again aren't offered for truth. I don't necessarily know that we should treat patients based on these relative risks, but the idea is that we can do a lot better than what we have for the CHA2DS2-VASc score. That we probably should be including some social determinants of health. We probably should be understanding synergistic risk factors now that we have electronic health records and all kinds of technologies to help generate these types of answers almost on the fly, live. But obviously, there's a lot of work to do there. So, so where are we going next? And I'm happy to talk a little bit about this. This is very preliminary, so I'm happy to talk a little bit about it in the Q&A. But we're looking forward to, we're going to collaborate hopefully with PCORnet, which is a part of PCORI is the Patient-Centered Outcomes Research Institute. And they administer a large clinical research mechanism across different sites in the country. So we participate as part of the Greater Plains Collaborative with a number of institutions. So essentially it could include not just our institution, but 10 other institutions and 17 million patients worth of data in this work. And then we also hope to include some very unique social determinants of health data, which again, I'm happy to go into more in the Q&A, to try to get at some of these unanswered questions around how do you resolve risk and understand some of the disparities and address some of them, potentially including data that are never, ever used in healthcare, at least not yet. So that's where we're going in the future. And that's kind of the scope of where we hope to move personalized risk stratification for stroke and atrial fibrillation. So I'm going to stop there. Happy to take any questions on that section. And actually, sorry, one more slide on the deliverable. So what does this look like in practice? And so this is a figure designed and replicates some of the work they've already done by Raquel, who's a really great MD PhD student working with Dr. Yandel. But it's essentially the idea that in the future, based on different factors, and these are not definitive, we could understand really at a patient level in our health system with real data at a very large scale, rather refined risk, patient to patient, year to year, and use that to drive decisions around treatment. For example, we generally think of an annual stroke risk of in the 1% to 2% range to warrant oral anticoagulation, and this could refine that much more than what we currently do. So obviously a prototype, a nice graphic, and hopefully where we're moving. So I'm going to switch gears at this point for a minute. And move on to another interest that's a little earlier in development on this campus, but hoping to generate some collaboration and some new ideas and some new science. And so this is the idea of understanding electrocardiograms at a much deeper level. I'll start a little bit with the background, which may be familiar to many of you, but the risk of the problem of sudden cardiac death, which is which among other things, in addition to being a dramatic events clinically, demonstrate a quite frustrating paradox, which is that, and this is nicely highlighted in a classic figure, but the paradox is that the patients of highest risk, so those with clear arrhythmia markers, prior myocardial infarction, those that have had a cardiac arrest, in general account for the minority of absolute events we see clinically in the world. And unfortunately, the majority of the events we see are in the general population. They may be high risk for events based on classic factors, but they have no other identifying feature that allows us to pick them out prior to their event. They're the events you're hearing about the news, the patients dropping dead on the basketball court, on the lacrosse fields, during the marathon, et cetera, et cetera. And this has represented really quite a frustrating challenge for many of us, obviously. And is in part related to the kind of multifactorial etiology of sudden death, which many of you are very familiar with. There are certain kind of structural diseases and triggers that we are familiar with and can identify and think about. There are genetic predispositions, some of which we can identify and many of which we probably cannot. And then there are more neurohormonal and autonomic influences, which many of you probably are more familiar with than I am, but drive a lot of this risk. And I won't go into more into the details, but I think many of you can appreciate that a lot of these events, all of them, have a terminal arrhythmia component driving them. Whether it's ischemia that creates repolar disease, whether it's a potassium channel defect, whether it's a drug that's impacting ion channel repolarization, but in general represents a terminal arrhythmia event. And so the question being, how can we predict those? And this has been a long-standing interest for many people. You guys are familiar with the 12-lead EKG. It, in fact, I want to point out, is all 12 leads across 10 seconds usually. We'll come back to this. But this is the classic 12-lead EKG that people have spent a lot of time trying to pick out markers of sudden death. I classify these generally as macro markers that you usually can see with the human eye. So they're markers of specific disease. Your QT is long, your QT is very short. You have a pattern characteristic of a syndrome, whether it's Wolff-Parkinson-White, Brugada, whatever it is. It's a morphology that a human can recognize. There are other changes that we recognize, whether it's classic ischemic changes or classic structural changes suggesting inherited arrhythmia syndromes that increase the risk of sudden death. These are not particularly novel. They're markers of clinical disease that are manifest on the EKG that humans can see. There are other more what I would call disease-independent findings, many of which still are based on human interpretation, but not all. So markers of repolarization, duration and dispersion, markers of depolarization as well, and then repolarization variance, based again either on human interpretation or machine. But these are all again based primarily on the image, and there's probably a lot more information in the EKG, just like in the prior example, there's a lot more information in the health record that we don't leverage for looking at this. And so what I think it comes down to is I think this is perhaps maybe a little more accurate kind of paradigm around sudden death, which is that there are many different factors. They probably, I believe, interact to a large extent with an underlying genetic susceptibility. And so the overarching kind of objective of what we're hoping to do is to leverage high fidelity, I'll talk about what we mean by this, EKGs that we believe contain way more diagnostic information that can be perceived by human interpretation or even kind of simple machine interpretation, and may add value for diagnosis, risk stratification, and management of these disease processes. And this isn't out of the blue. I'll talk about some prior work with some collaborators, but just so we're all on the same page, and this is probably review for a lot of you, but just in case, the classic 12-lead EKG has 6 limb leads, 6 precordial leads. 4 of them tend to be mathematically independent, and so for the purposes of this work, most people focus on 8 leads only. And the picture that I showed with all those leads, gets transformed to numerical raw data of the recording. And so this is what it looks like in base 16, not obviously terribly interpretable by us, but essentially is 8 leads at 500 hertz sample across 10 seconds. So you get a matrix of 5,000 points, and then distribute over 8 spatial leads. And this is a paradigm of what's been done and what we're hoping to kind of not so much replicate with, but build on through some collaborations on campus. But essentially, and this is a figure from the group at Mayo, which you'll see a lot of citations from. They've been great collaborators and have done the bulk of the work in this field so far. But essentially, they take the separate leads and build them into a machine learning convolutional matrix where they identify, where they train the model to identify a specific output or a target variable. And so this is what we mean by kind of machine learning or artificial intelligence on EKG, at least for the purposes of this talk, and where I believe there is a lot more information contained in the diagnostic than we appreciate visually. And why do I think this is so promising? So, so this is kind of the furthest along we are. We're actually hoping to maybe bring this to our campus, depending on the collaboration and the IT resources. But this is kind of down the road what Mayo has done. And so they've created a dashboard in their EHR where the algorithm that they've developed can take an EKG and give an age estimate, give an estimate of whether that patient has a low ejection fraction, give an estimate of whether that patient has hypertrophic cardiomyopathy, and there's a recent paper from the Lancet, like last 2 weeks, on predicting atrial fibrillation. And so this is obviously, ideally where we go in the future. And it's a busy field. You'll notice lots of different groups interested in this. You'll notice numbers of patients are relatively large, although not all of them. But we're often looking at tens of thousands, if not more, EKGs, in a variety of different outcomes, and I'll show more. But it's a very, there's a lot of interest in this field. And rightfully so. So there are papers published, again, a lot by the Mayo Group and some others that have demonstrated algorithms with promise, not guaranteed, but with promise to be able to dissect out sex, different demographics, risk of arrhythmia, either that is silent and pre-existing that you don't see, or that may develop in the future. The ejection fraction, which I believe they are actually either in the process or have gotten FDA clearance for their algorithm for ejection fraction. We helped the Mayo Clinic with that. Disease states throughout the heart, and then even a paper that demonstrated promise for an algorithm that detected liver cirrhosis based on the EKG. I'm not going to go into the details here, and obviously there are issues of external validity, replicability, explainability, but I think this list at least demonstrates the promise of what we could do with these data. And so where are we going on our campus, or where do I hope we're going? So at University of Utah, we have invested in the technology to extract out those raw data for all the EKGs in our health system. Right now, it's about 1.2 million, but it's an ongoing process. And we do about somewhere between 200 and 400 EKGs a day in our health system, at least based on how many I get to read. We, on this campus, have some singular additional data that many other health systems don't have. So in addition to the raw waveforms, we have all of our patients' genetic testing results in a coded fashion through one vendor. And we have pretty unique genealogy and familial relationships through the Utah Population Health Database that could be leveraged for either sampling of patients and risk stratifications or recruitment or understanding genetic relationships based on the EKG. And then we have particular expertise on our campus through this group, obviously, as well as SKI and our bioinformatics groups in terms of analytics and computation. And so that's where I see the promise for this technology. So how does this relate to sudden death, and what can we do? What can we do in sudden death?
You can always put 30 minutes on my schedule.
Yeah. Oh, okay.
And
So
Greg, you're unmuted.
So a lot of the algorithms I've shown so far have used what's called labeled data. So you have a cohort of 40,000 echocardiograms, you have 40,000 EKGs, a proportion of them you use to train an algorithm and you test it on the result. And you use a fairly large group to train the sophisticated algorithms. That's great for things that we have collected routinely and well-defined: age, sex, ejection fraction, future AFib, those sorts of things where the data are widely available, the definitions are easily agreed upon, and you can replicate in other health systems relatively easily. Sudden cardiac death, among other clinical etiologies, presents a challenge. It's relatively rare, fortunately, but not rare, so rare that we shouldn't study it. It can be difficult to identify or to define, and it's often not fully labeled. And that's also true for kind of underlying genetic predisposition. Not everybody has genetic testing. Not every genetic defect is actually defined. Uncle Sam died in his sleep. It was a massive heart attack. Was that sudden death? So-and-so had a single car accident, in the middle of the day. What was that? Et cetera, et cetera. So sudden death represents a challenge, like many others, at getting at this. So what have we started to do here? And this is really great work done by a third-year medical student, actually, James Brundage, working with some people here as well as at SCI, trying to understand and trying to kind of move some of these methods forward on our campus. And so we started with the data set that we collaborated with the Mayo Clinic on, and basically did a trial of a feasibility study and looked at what we had and what we could use. And so in our system, we have, again, about 1.2 million EKGs of about over 400,000 unique patients. We can match about 380,000 to ejection fraction data. We furthermore have to limit it to patients with an ECG that was prior, although you could argue about a time frame there, within 30 days. And we still get to a healthy population, although not quite a million, of 60,000 EKGs and 39,000 patients. And in a data set, overall, once all the data settles, and once we have all the other fields that we might want, and I'll talk about that, of a pretty good data set. And this is, again, just more of a proof of concept in understanding what's feasible and how we can replicate Mayo methods, maybe even improve on them, et cetera, et cetera. So 40,000 EKGs and 25,000. So I'll skip forward. James did some great work validating and replicating some of the work that Mayo has done looking at ejection fraction. And then we wanted to kind of try to look at how would we tackle, for example, genetic defects. So if we have 1,000 patients that have genetic testing, some are positive, some are negative, how do we use that data to look at a much broader data set of EKGs where the patients don't have genetic testing? And so James and some collaborators at SCI, who are, again, much more smart than I am, looked at what's called non-contrastive self-supervised learning, or essentially training an algorithm to identify the labels for us. And what that means is, so in supervised learning is what I talked about earlier. You have a labeled data set where the outcome or the target is known. You take a proportion of it, you train the algorithms to identify that, and then you test it on the remaining portion by removing the labels and testing how the algorithm does. You have a fully labeled data set. So that's supervised. And again, this is the electrophysiologist understanding machine learning, not the computer scientists or electrical engineers understanding. In this specific method of non-contrastive self-supervised learning, the goal is to identify features that may be important for understanding the target outcome based on labeling that you already know. So the example that we used, that James employed, was we said we have 40,000 EKGs. Let's pretend we don't know the ejection fraction for most of them, but we do know sex for most of them. So can we use the target variable of sex to identify features in what we term cheaply labeled data, so a data set with labels that are relatively easy to come by? And can we use the features identified based on, for example, sex, to train an algorithm to identify ejection fraction in a much smaller training cohort? So say you only have 1,000 patients with ejection fraction, or 500. Can you use what you know about sex in 40,000 to refine an algorithm on a much smaller cohort. So for example, if I have 800 patients with genetic testing results, but I have 1.2 million EKGs, can I understand features of the EKG in those 1.2 million that identify markers of sex or age or whatever it is, and apply those features to understanding what might identify genetic defect in those 800? Because as I showed that big blue table, 800 patients is not a big cohort to do this sort of work, unless you have some fancier models. And so the next slide is very preliminary data that James has presented, and we're hoping to, I think he's presenting at a meeting, but essentially demonstrating sex-based initialization can improve on algorithm performance in partially labeled data set using smaller training data sets. And again, these are very preliminary. To his credit, James has done great work with some mentorship over at SCI, and I think shows some promise for what we could do and what we hope to do with some collaborations. And so where do I hope this goes in the future, and certainly welcome collaborations on this front, is I think there are potentially two opportunities. There's probably many more, but where I see first is we have a pretty unique infrastructure here and ability to get genetic testing and familial relationships. And I believe that offers really a unique opportunity to understand genetic markers or underlying genetic predisposition to these events that we don't otherwise identify. Either genetic testings don't pick them up, we don't see them by EKG. And so can we use these self-supervised learning techniques to understand underappreciated markers of underlying risk on EKGs using very small labeled data sets, relatively speaking, and very large unlabeled data sets? So again, as an example, 800 patients with test results versus 1.2 million patients in the system. And we potentially could understand underlying risk, a surrogate for underlying risk through genetic risk. So if we had an algorithm that, for example, identified a potassium channel defect that resulted in a genetic test, those are the types of markers we could look at. I think there's also an opportunity through the UPDB to understand actual cases of sudden cardiac death. And there's some other ways of identifying those events clinically in the record, and more sophisticated methodology that I didn't touch on today, where we could train on a very large data set to predict those types of events based on EKG. And so this is where I hope we're going, and I kind of welcome discussion and potential collaboration on these topics. And we're in the process of trying to secure some funding for this as well. So with that, I will give a bit of a summary. I look forward to kind of Q&A and then elaborating a little bit more at that time. But I think in summary, hopefully I've conveyed to you that there are still major challenges in risk stratification among patients with arrhythmia disorders. That probably doesn't come as a surprise. I think we need to focus more on deriving valuable information from what we already have than developing fancier tests and more expensive diagnostics necessarily, at least in some of these spaces, especially given the universal prevalence of electronic health records, for one, as well as digital diagnostics. And so I think there are some really novel approaches, particularly on this campus, for leveraging these existing data to improve care and risk stratification in these patients. As I mentioned at the top, I've been really some brilliant collaborators. So on the AFib side, Ian group, as well as the data warehouse. Again, Raquel is an MD PhD student who was instrumental in a lot of in that grant and a lot of this work. And then on the ECG side, again, James, a student, and then Bao and Tolga over at SCI, who mentored a lot of that. Rashmi Shah, who was in our division, laid a lot of the groundwork for this. And then a lot of the data warehouse people that really did a lot of heavy lifting just to get the data we need to run some of these experiments. So with that, I welcome any questions.